I want to thank the organizers for the kind invitation to allow me to share with you this morning our gene therapy strategy to prevent or treat neurodegenerative diseases. I'm sure some of you, have relatives or friends that had understanding of how devastating this disease could be. And so what my team would like to do is to prevent this in the future. And so our target is really to address this loss of function of an RNA-binding protein, I'm going to tell you more about, that underpin these major neurodegenerative diseases. So I'm a professor here at Johns Hopkins in the School of Medicine. I've been here in the Department of Pathology and Neuroscience for 3 decades, and I've been focusing on these neurodegenerative diseases to date. And the most exciting comes from this work I'm going to tell you that started about 15 years ago. And my colleague, Jonathan Lang, he's also a professor in my department. And he's actually the one that discovered this function of TDP-43 that we're going to be discussing today. And so as I indicated, loss of function of TDP-43 really underpins now neurodegenerative diseases, ALS, FTD, Alzheimer's disease, and even non-neurological diseases such as inclusion body myositis. The understanding of this basic mechanism that's involved in these diseases really point us to the appropriate therapeutic strategy that we now could move forward and bring to the clinic. One of the major hurdles that we face over this past decade is that we didn't know how to detect patients that have loss of this TDP-43 function. But now we do. So I could tell you a little bit about that. And so given the prevalence of these neurodegenerative diseases, we estimate the burden and the potential size of this market is in the order of $12 billion. So what is TDP-43? So it's an RNA-binding protein that plays a role in RNA splicing, a role that we never thought about before. We like to think of it as a guardian of the transcriptome. So across all 20,000 RNA, there are actually these so-called cryptic exons that are hidden within long intronic element of your gene, of all your RNA. And if some of these cryptic exons get spliced into the normal RNA, you would disrupt the gene expression because they introduce premature stop codon and kill the RNA. And so the cell have figured a way to protect itself. And it's by these RNA-binding protein, TDP-43. It binds to these regions where the cryptic exon resides and makes sure and tell the spliceosome, don't splice here. So that's how they could prevent these cryptic exon inclusion. You make a normal transcriptome. But in disease, what happened is TDP-43 gets clear from the nuclear compartment, so they're no longer able to perform this function. And therefore, you totally disrupt the transcriptome, and that is the problem underlying neuron death in neurodegenerative diseases. And you can clearly demonstrate this happening not just in mouse model and cells, but actually in human disease that have ALS or FTD or Alzheimer's disease. And the loss of this function, now we can clearly know, could happen very early on, in early stage disease, even pre-symptomatically. So that is a huge bonus for us, because now what we can do with the biomarkers is we can recruit patients as early as possible for the clinic for these type of therapies. So this is the idea that we have. With the biomarker, we can actually recruit patients at the prodromal phase of the disease and deliver, using this AAV, this splicing repressor, to complement the loss of TDP-43. So our idea is that when you lose TDP-43 function, there's no compensatory mechanism. You lose it, that's it. So our idea is to sort of immunize our nerve cells, provide second backup strategy. In case you lose its function, you can protect the neuron. You prevent the inclusion of cryptic exon, and you protect neuronal death. So over the last 10, 15 years, we've actually demonstrated the loss of function of TDP-43 is critical for neuron loss. We have a biomarker that can recruit patient at the earliest time possible, and we established this modality, AAV, adeno-associated virus, to deliver this payload to complement the loss of TDP-43. And most recently, we show indeed loss of TDP-43 really drives neurodegeneration in Alzheimer's disease. So let me just give you a very quick example how this might work in model systems. So you can see a mouse that is paralyzed, you can, on the left there, because it loses TDP-43 function. But if we introduce this drug to these mice, symptomatic even, they protect these mice. They no longer die of ALS, motor neuron disease. This is a spectacular finding. Delivering pre-symptomatic to rescue such a robust phenotype. You can see the survival. None of these mice so far die. All of the treated with the control, they have passed away. The reason is we can protect, using our modality, 70% of these motor neurons. And so our goal is to really focus on the basic mechanism of TDP-43 pathophysiology. Unlike other companies who might be targeting the aggregation of TDP-43, or even some of the downstream target of TDP-43, loss of function, because they do not address the basic fundamental mechanism of disease. And our strategy does. And so, in summary, what I just told you is that we really have developed a robust therapeutic strategy that's validated in model systems, accompanied by a pre-symptomatic biomarker that will help us recruit patients early and use it as a target engagement to show that our therapy actually works. And so our next step is really to get funding, seed funding for our new company to move our effort towards clinical phase 1 trials. And so if you're interested in this work, please come and talk to me anytime throughout the day or email me or Jonathan. Thank you very much for your attention.